Good morning and welcome to today's Updates in the Research and Care webinar, spotlighting new research in neuromuscular diseases. We are glad to have you join us. My name is Tess Taylor and I'm a specialist with MDA's community education team. Today's learning event is part of our larger MDA community education program, which focuses on bringing the neuromuscular disease community together around education and resources. Be sure to visit the community education section on mda.org for a complete listing of all of our programs and educational materials. We are recording today's event and we'll be posting it to the website for on-demand viewing in just a few weeks. Everyone registered for today's session will receive a link to the recording via email when it is ready. Please know that all phone lines have been muted, but you can still ask questions. As participants, we have a Q&A session at the end of each speaker's presentation. Simply use the chat feature to type in your question, and feel free to type your questions and send them in as you think of them, and they'll be addressed after each presentation. Please note that we might not be able to answer questions that are highly specific to your exact situation. We encourage that you contact your healthcare provider as they know your situation best. We'd like to thank the generous sponsor of today's program, Biogen. Their support helps make programs like these possible. MDA's mission is to help children and adults living with neuromuscular disease live longer, stronger, and with more independence. Through targeted programs and innovative initiatives, we address the most pressing needs of our community. This webinar series is designed to provide timely, transparent, and accessible communication about scientific advances, clinical trials, and MDA-supported research. This will be a meaningful opportunity to connect between researchers and the MDA community. And today we're joined by a lovely panel of presenters. I would now like to turn our time over to Dr. Brian Lin, who is MDA's Research Portfolio Director, and then he will guide us through our presentations today. Hi, Dr. Lin. Thank you so much for being here. I will now go ahead and turn the time over to you.
Perfect. Thanks, Tess. Let me share my screen.
You can go ahead and do that, and you'll kick me off, and that's great.
Okay, perfect. Can you see that?
Yep, looks perfect.
Okay. So hi, everyone. So welcome to this new research series that we just started this year, as Tess mentioned, with a really nice introduction. My name is Brian Lin. I'm the Research Portfolio Director at the Muscular Dystrophy Association as part of the MDA research team. And so throughout this new research series and in future webinars, you'll hear more from me, and you'll also hear from Dr. Angela Leck, who is our Chief Research Officer and who leads our research team. And we'll be joined throughout the year by many scientific experts from across our MDA and neuromuscular research community. And really the purpose is to bring you insights into the latest science that is happening. And so we really started this research webinar series with the goal of creating a forum where you can connect, you can ask questions, and really learn directly from the researchers who are at the front lines of developing those new treatments, conducting really impactful scientific studies, and running those clinical trials that are happening right now. And they're also just an amazing source of information about the latest scientific advances. And so tentatively, these webinars will occur every other month, and they'll focus on really timely and relevant topics. And so these may shift depending on developments that are occurring in the field. But some of the topics that we have planned are shown here. And it includes, for example, genetic medicines and muscle regeneration, some topics that the community has shown a lot of interest in. And these are really areas that are highly relevant now, and they will be for many years to come. And so throughout these webinars, you'll hear about specific diseases as well. And I think it's important to keep in mind that although a particular disease may not be relevant directly for you, the research, the techniques, the new technologies that are being used as part of that research are relevant across all the neuromuscular diseases. And so, something exciting that you might hear that's happening in DMD, for example, may end up being applied for other diseases like LGMD or FSHD down the road. And so today we're going to spotlight new research that we're funding starting this year. You'll hear directly from those investigators who recently received those awards, get a look at the research that's happening in their lab. You'll get a preview of what's ahead in their MDA-funded projects as well. And just before we get into that research, for this one slide, I just want to really briefly highlight our research grant program that is supporting some of the studies that you hear about today, and also answer a very common question that we received from our MDA community, which is how do we actually fund grants? How did we decide to pick these particular projects over others? Because as you know, MDA, we are an umbrella organization. We cover over 300 different neuromuscular diseases, and we really can't possibly fund all of those diseases at once. And so we have a really rigorous process that's designed to identify the strongest, the most impactful, the most innovative projects that will receive our multi-year supports. And so really this is how it works. We typically start with an open call for applications. And so these are investigator-initiated applications. They submit their projects that they're interested in getting funding for. We don't prioritize on our end based on disease or where they are, what institute they're at, or a particular funding area as well. And so these applications are then reviewed very carefully by our research advisory committee that consists of over 50 scientific experts, and we'll often get additional experts on our committee for that particular year because of particular expertise that's needed for a grant application. And the projects will be scored, and they'll be scored on the scientific merits, the innovation, the impact. And so the best projects will really rise to the top. And so these are the projects that we end up funding because they have the greatest potential to make a real difference across the neuromuscular disease field. And we've seen on many occasions that successful projects and advances for one particular disease can really influence and set the stage for progress in others. And so we focus on funding the best science that's possible. And so in 2026, this is just a look at our research portfolio. We are actively supporting over 85 exciting projects across the field. And this distribution of diseases can really change year to year based on what projects and applications we end up funding based on what's submitted and whatever scores the highest. And so for this first webinar, the 3 new additions to this portfolio that you hear about today are by these great investigators. We have Dr. Charlotte Sumner with us, Dr. Chad Heatwole, and Dr. Matthew Nolan. And kicking us off will be Dr. Charlotte Sumner. And Dr. Sumner is a professor of neurology, neuroscience, and genetic medicine at Johns Hopkins University School of Medicine. She co-directs the Johns Hopkins MDA Care Center, the SMA and CMT clinics as well. And her lab focuses on motor neuron and peripheral nerve diseases. And so welcome, Dr. Sumner. We're so happy to have you with us.
Well, thank you so much, Brian. I'm just going to share my slides. Boy, I'm struggling a little bit here. Why is that? Oh. I had this working before. I'm going to stop for a sec. Excuse me. Try one more time. Okay, I think I got it this time. Yes?
Yep, there we go. Perfect.
Okay, well, thank you guys, everyone who's on. Thank you to the MDA. Thank you for listening. I'm going to talk to you a little bit about the work that we've been doing to try and advance treatment for particular genetic neuromuscular diseases. And I'm going to tell you a little bit about that today. This story really starts some time ago. This starts back in 2010 when our group, as well as 2 other groups, identified that mutations of a gene that we abbreviate TRPV4, or transient receptor potential vanilloid 4, cause Charcot-Marie-Tooth disease type 2C, as well as forms of distal spinal muscular atrophy. And these 3 studies were published in this journal, Nature Genetics. And this is just a photograph of one of the first individuals that I met with this disease. And, it's a pretty severe disease in many cases. Individuals living with TRPV4 mutations often develop quite severe weakness of limb muscles, the vocal fold muscles, and the diaphragm. And as you can see in this image, Kelly here is quite weak, is using a wheelchair, and has a tracheostomy because breathing is difficult for her. You probably never heard this term, TRPV4, and so I thought it might be helpful just to mention what kind of protein it encodes. TRPV4 is a type of ion channel, and ion channels are pore-forming proteins that sit within membranes, often the cell surface membrane, and they allow ions to pass across a membrane. And ions are molecules that have a net electric charge. So ions you've heard of include sodium or potassium. Another one would be calcium. And so these ion channels often sit in the cell surface membrane and allow these ions to pass across the cell surface. And often they can be closed or open depending on stimuli that can activate channels. So TRPV4 is one of these ion channels, and it's part of a superfamily of ion channels called the TRP channel superfamily. And in fact, the Nobel Prize was awarded a few years ago to David Julius, who discovered these channels. So they're actually more recently discovered family of ion channels. And this is what the ion channel looks like here on the right side. You can see that TRPV4 sits, again, within the cell surface membrane. It has 4 subunits, and it mainly passes positively charged ions, or what we call cations. And in particular, it fluxes calcium. And when activated, this TRPV4 channel opens and allows calcium to pass from outside the cell to the inside of the cell. And the thing about ion channels that's useful is that in addition to playing these essential physiologic roles in cells, we know that we're very good at developing drugs that can modulate the activity of ion channels. So we're very good at developing drugs that can activate an ion channel or inhibit an ion channel. And many of these medicines we use today in clinical practice. For example, we use medicines that block ion channels to treat, for example, problems with heart rhythm, for example. So this channel, TRPV4, that we and others discovered when mutated can cause neuromuscular disease. When we first identified that mutation, it seemed likely that maybe we could target this protein therapeutically with drugs that would modulate the channel activity. But before testing a drug, we had to understand how do the mutations that we identified in patients actually affect the function of the TRPV4 channel. And we can do this, we can do functional testing in cells. These are cultured cells. This work was done by Brett McRae in my lab. And on the left side of these images are cells expressing the normal wild type channel, but on the right side are cells expressing the mutant channel. And what you see in these videos are the flux of calcium into the cell. And you can see with cells expressing the normal channel that there is some influx of calcium. But when you express the mutant channel, you can see that these cells become very, very red, indicating an increase of calcium influx into the cell. So these kinds of studies and a lot of other work taught us that the mutations that are causing neuromuscular disease are really causing the channel to be overly active. The channel gets to the cell surface normally, but when it's there, it fluxes too much calcium into the cell. And this suggested that if we could inhibit the channel, that this could be beneficial to patients with the disease. So in order to further explore whether or not inhibiting the TRPV4, the mutant TRPV4 channel would be helpful to patients with these mutations, we built a mouse model of the disease. And we did this by inducing a single change into the DNA of the mouse TRPV4 gene. And when we did that, we did see that the mice got quite sick. This is a survival curve. Mice that don't express the mutation live a couple of years, but mice that were carrying the mutation in the mouse TRPV4 gene died by about 3 or 4 weeks of life. And they showed weakness of their neck muscles and of their forelimb muscles. And when we looked in the mouse at the motor neurons in the spinal cord, we could see that the number of motor neurons is reduced, and their axons, the wire that connects the cell body of the neuron to the muscle, was actively degenerating. So this allowed really and showed us that if we were able to generate a mouse model that expressed a mutant form of the TRPV4 channel, that did cause quite significant disease in the mice with degeneration of motor neurons and weakness of the mouse with reduced survival. We wanted to understand in that mouse how the mutant protein was causing disease. And what we set out to do was to understand where the TRPV4 channel is actually expressed within the nervous system of the mice. This is a cross-section of the spinal cord where you can see motor neurons extending out to muscles. And when we looked in the spinal cord here in cross-sections similar to this, we actually did not see that TRPV4 was expressed in the motor neuron itself. In fact, where TRPV4 is expressed is within the blood vessels, not within the neurons directly. The spinal cord, like all tissues in our body, has to get blood supply via blood vessels that branch out across the spinal cord and provide blood supply to neurons as well as the other cell types of the spinal cord. But in the nervous system, the vasculature is even more important in some ways than in other places because the nervous system is protected by what are called blood-brain barriers or blood-spinal cord barriers. And these barriers prevent proteins from passing from the vasculature into the CNS, the spinal cord and brain, because some of those proteins, when they leak into the nervous system, can be toxic to neurons. And so this raised the possibility that TRPV4 expression within these endothelial cells of the blood vessels of the nervous system was actually disrupting this protective barrier between the blood supply and the nervous system, and that proteins were leaking from the blood into the nervous system and causing disease. And indeed, when we look in the spinal cord and inject a dye here called easy-biotin, in normal mice, there is no leakage of the dye into the nervous system, and this is what we would expect. But in the mutant mice, we see leakage of blood products into the nervous system. And we believe it's this breakdown of these endothelial cells and leakage of blood products into the nervous system that causes the neurons to degenerate. Now, to prove that, we actually removed TRPV4 from individual cell types in the nervous system, including from endothelial cells. And we proved, in fact, that the only cell type that seems to trigger disease is the endothelial cells. When we removed mutant TRPV4 from neurons or from glial cells, there was no effect on the phenotype of the mice. They still got sick. But when we removed the mutant TRPV4 from endothelial cells, this completely prevented the mice from getting sick. Mice that had the mutant channel into endothelial cells all died, but mice that did not all survived and did not develop any weakness. So in this disorder, surprisingly, it appears to be that it's the vascular endothelial cells that are really triggering disease. And so it raised the question, could we target that ion channel in endothelial cells with a drug that actually inhibits the channel? And we initially did this with a drug that was available from GSK. We treated mice with a drug that inhibits the TRPV4 channel, and we started drug at a time when the mice are already sick, and we call this stage B. When we did that, mice that would have all died by 3 weeks of life are now living a completely normal lifespan when we treat with a high dose here in the blue. And also, whereas mice would develop problems with weakness, when we treat with the antagonist, the mice are completely restored in their strength. And importantly, when we look at the leakage of blood products into the spinal cord, we see that that is completely reversed when we treat with a TRPV4 antagonist. So this is really exciting and promising data from mice that perhaps a TRPV4 antagonist could be helpful to patients. But we still have a lot of work we need to do. We're fortunate now that we're partnering with a company called Actio Biosciences, who has developed a novel TRPV4 antagonist, and they are planning clinical trials in patients with this antagonist. But we've really needed to understand the human disease much better than we do now. And so at Johns Hopkins, we've been doing an ongoing natural history study in individuals with the TRPV4 mutations. And importantly, in addition to support from Actio to do this study, we've been receiving support from the MDA. And we are seeing many patients over time and really trying to evaluate them with multiple different measures that we think will be used during a clinical trial of the Actio inhibitor. In addition, and really the topic of the recently funded grant, is we're doing more work in mouse models to really make sure that we're ready for clinical trials in patients. So we've built another mouse, what we call a humanized mouse model of TRPV4. I mentioned that the last mouse is the mouse gene with a human mutation, but in this mouse, we're actually expressing the full human gene and making the full human protein. And this is really important to really verify that the human channel causes disease and that the human channel can be blocked with the small molecule antagonists and ameliorate disease. So those studies are ongoing with the help of MDA support. We also are investigating in this grant whether the channel expression in only endothelial cells is sufficient to cause disease. We know it's necessary, but is it sufficient? Or do other cell types actually play very important roles in generating the disease phenotype? And this is relevant to therapeutics because the drug that we're using will really primarily target the endothelial cell. But if other cell types are really important for the disease, that drug that we're currently using may not be sufficient. And then finally, the last goal of the grant is really to try and identify blood-based disease biomarkers that we can use during a clinical trial to teach us whether the drug is sufficiently engaging the TRPV4 channel and suppressing it, but also of whether they are good biomarkers of disease activity. And some of you may have heard about an emerging blood biomarker that we're using in lots of different neuromuscular disease called neurofilament light chain in this case. And you can see here that in our mutant mice, the neurofilament is elevated in diseased mice. In fact, that elevation rises with disease during disease progression. And we are testing whether the TRPV4 antagonists can reduce neurofilament levels, and that will give us proof of concept that that also may be something that we can use during the clinical trials. So I'm going to stop there and just thank the people from my own group and key collaborators who have been central in all the work that I've presented. A huge thanks goes to MDA. MDA has been absolutely central in the development of this work, both funding the very earliest development of models so that we could study this disease, supporting the natural history study of patients, and now really key, very translationally relevant studies that will help us do clinical trials in the best way that we can. And I'm happy to try and take any questions if there's still time.
Thank you, Dr. Sumner. That was a great presentation about your research. Tess, do we have any questions for Dr. Sumner from the audience?
No specific questions for Dr. Sumner at this time, but I agree with Dr. Lin. That was such a wonderful and fascinating presentation. Thank you so much for joining us.
Of course.
Okay, so next presenter is Dr. Chad Heatwole. Dr. Heatwole is a professor of neurology and director of the Center for Health and Technology at the University of Rochester Medical Center. He leads an internationally recognized research focused on multi-center therapeutic studies for rare diseases and the development of sensitive patient-relevant outcome measures for clinical trials. He's also a practicing clinician, so he sees patients and families with muscular dystrophy in his neuromuscular clinic. And so welcome, Dr. Heatwole. Thanks for being here.
Hi, Brian. Hello, everybody. Great to be here with you. It's a little bit of a homecoming for me. I've been working with the MDA for over 20 years now, and it's just been a wonderful relationship and really has been a joy to kind of advance clinical research for the benefit of patients through this partnership. And today I'll be talking a little bit about our work and what we do at the University of Rochester. And specifically our upcoming new grant called Leopard DM2, which is longitudinal endpoint optimization to provide assessment of relevant drugs in myotonic dystrophy type 2 patients. Ooh, okay. So just disclosures really have, like I said, had a lot of nice collaborative relationships over the years and been blessed with that. To really help a lot of different organizations and a lot of different patients with rare neurologic disease and especially neuromuscular disease. My background is I'm a clinical neurologist, but my true joy is really working in the neuromuscular field, and I run a neuromuscular clinic and also focus a lot of my research in that area. And I also head an entity called the Center for Health and Technology, or CHET, again at the University of Rochester. And it's a 100-person organization that's really designed to advance human therapeutics, health, and knowledge through exceptional people, skillful research, and partnerships. And we work out of an academic setting, but we have a lot of connections outside of academic settings, specifically with the FDA, patient groups, foundations, organizations such as the MDA, NIH, philanthropy, government, and industry. And there's 6 organizations out of CHET, and they're all focused for therapeutic research, late therapeutic research for neurologic diseases. We run a clinical trials coordination center where we run multi-center clinical trials worldwide. We have a clinical materials service unit that ships drugs for clinical trials. We have a CHET analytics group that does deep dives into analytics and predictive modeling to help clinical trial design. CHET innovation is novel outcome measures and devices like Apple Watches to measure function. CHET outcomes is what I'm going to talk about mostly here is the development of patient-reported outcome measures to give patients an insight and input into whether a therapy is effective or not. And CHET Health is getting our devices out into the community and getting the community into our studies. So altogether, we've run over 155 clinical trials. These have led to 12 FDA drug approvals, 45,000 research patients. We've developed over 250 disease-specific outcome measures, and we've been operating for over 30 years. These are our FDA approvals. Most of them have been in the movement disease space, but we do also work in neuromuscular space, and that is my area of subspecialty, and have a FDA approval for primary hypokalemic periodic paralysis. So our outcomes group is really designed to develop highly specific, state-of-the-art, disruptive, patient-reported outcome measures to give patients a mechanism to quantify how their disease is and how their disease burden is in the context of a clinical trial. And our group really set forth to do this the right way, make highly sensitive, very meaningful outcome measures, really for use in therapeutic trials. And we've had a lot of success of that over the last 20 years. And unlike a lot of patient-reported outcome measures that live in the community, we develop ours using extensive research from the ground up to fully satisfy regulatory and FDA criteria for use as effective measures to support drug labeling claims in clinical trials. And so there's a lot of work that goes into this, and to do it right and to satisfy all those criteria, here are 9 things that the FDA requires. I won't go through all of these, but they have to do with reliability and meaningfulness and content validity and statistical validity, and limited patient burden and low redundancy. And when I say validity, there's actually 7 different types of validity that really need to be shown in a really good outcome measure. Again, I won't go through these in this context of this talk, but it's difficult to do, and our organization really does a good job in documenting and ensuring that all of these things are met. And there's many different advantages to our instruments compared to generic instruments that aren't for a specific population. And our disease specific, they're very relevant to the patient. So it's important to ask questions that are important to specific populations. And so we do that. They have lower disease or lower burden to patients to complete, because what really generates burden is when you ask questions that are irrelevant to a population. They have strong correlation to functional capability. They can be used to lower the sample size and add efficiency to clinical trials. They can be used to emphasize relevant symptomatic themes, increase sensitivity to detect meaningful changes. And so if you have a good therapy and the wrong outcomes that aren't able to detect meaningful change in a patient's state, your study is going to fail. And so it's really important to get these outcomes right. And they're better suited to measure disease progression over time. And we have a large group, again, about 100 people that works on these: patient and caregivers, outcomes researchers, biostatisticians, disease-specific experts, advocates, psychometric experts, and epidemiologists. There's a long process, but we heavily rely on patient input through interviews and cross-sectional studies. And then we do beta testing on our instruments and statistical testing. And we do small longitudinal study and then long longitudinal studies, which I'll talk about shortly. And this is a partial list of the instruments that we've developed. The one that the Leopard DM2 study is really going to be focused on is for myotonic dystrophy type 2. But as you can see, we have a lot of conditions that the MDA is interested in here: myotonic dystrophy type 1, FSHD, Duchenne muscular dystrophy, limb-girdle muscular dystrophy, and we have state-of-the-art disease-specific outcome measures for all of those diseases that are really revolutionizing how studies are being done. In fact, they're being used in over 100 studies worldwide, and 41 validation studies or translations have been done. So our instruments are being used in 41 different countries. And they're multifactorial. And so a patient can complete these instruments in about 10 minutes. And then they get a score from 0 to 100 in all of the symptomatic areas that are most important to that disease. And so it might be mobility and pain and fatigue and social health. And it just depends on which disease and which particular instrument. And what we found is that our instruments are widely preferred over existing patient-reported outcome measures. This was a study we published in Muscle and Nerve in 18. showing that our first instrument, the Myotonic Dystrophy Health Index, was preferred over INQOL and SF36 by patients. Despite being slightly longer, it's preferred by patients because, again, it's asking questions that are relevant. And the format is really important. So the way our format of our instrument is, patients can go through the questions very rapidly. In fact, they can go through 8.3 questions per minute. And that's just because the format's very easy on the eyes and easy to use. And that's in comparison compared to other instruments that are commonly used in studies like the SF36, that only 3.6 questions per minute, and things like the NeuroQual, which is only 1.9 questions per minute. And our studies have been really responsive to detect therapeutic gain in industry trials. And this is an example of just 5 studies over the last 24 months. In the DINE study, our MD high detect therapeutic gain over traditional measures in response to their antisense oligonucleotide. Neurocrine had a study for Huntington's disease and for valbenazine, and our instrument detected therapeutic gain, and that drug actually got a approval through the FDA. Spinraza has been the effects of Spinraza in adults has been detected by Craig Zeidman's group in the SMA high. One of our instruments detected therapeutic gain in response to that drug prior to any of the functional measures. Applied Therapeutics works in CMT. Our CMT high did good work there. And then FSHD or FSHD high detect therapeutic gain in our local starfish study. So for myotonic dystrophy type 2, we started out with patient interviews. Hey, what's important to you? What needs to be in one of these patient-reported outcome measures? We gathered 943 quotes, identified potential symptoms of importance, but just because someone says it doesn't mean it's important. So we ran a large cross-sectional study with 74 patients, over 12,000 survey responses to get the prevalence and relative importance of specific items in myotonic dystrophy. We published this data in Neurology. And then we take that data and we pick the questions that are most important to patients, the ones that have the strongest ability to detect therapeutic response in a clinical trial, and also statistical power, things that are not too vague, that have easy language to understand, and not abrasive or redundant. And there's a whole process that we go. So we build this instrument, then we do statistical analysis, then we do beta testing with patients to make sure it's really acceptable. Then we do reliability testing and to make the final instrument. And so for myotonic dystrophy type 2 instrument, here's an example of its power. So every time a patient completes it, they get a score on each one of those individual subscales at the bottom. And so those are subscales that are really important to myotonic dystrophy type 2. Mobility, hip, thigh, and strength, and knee strength, back, chest, and abdominal strength, shoulder and arm function, hand function, emotional cognition, sleep, fatigue, pain, myotonia, GI health, swallowing, activity, participation, communication, social satisfaction, breathing, and then there's a composite. And so what you see in this graph is that, versus those that are on disability versus those that are employed. Those subscales, also independently validated the total score, can differentiate between those with higher or lower disease burden. Higher scores, higher disease burden in this case. And also for the total score, we see that patients with less education, that are on disability, that are female, and that are older have higher levels of disease burden compared to their alternative groups. And we've published the data on the validation of this instrument. And so now we've come to the point where it's really essential and important to look at the longitudinal performance of this instrument and the ability of this instrument to detect disease progression in myotonic dystrophy type 2 patients. And this is the LEOPARD 2 study. And so we're going to recruit, in partnership with the MDA, 120 patients with DM2, and we're going to follow them in a longitudinal remote study over a 30-month period. And we're going to gather longitudinal data about DM2 using our instruments and other instruments. And we're going to identify factors that are in this population that are associated with a faster, slower disease burden. This will give us insight, a clinical insight. Maybe there's factors that really can prevent the progression of this disease or associated with a slower progression of the disease. And this data will also be critical in optimizing the final instrument, really determining patient preference for outcome measures going forward, and developing key longitudinal metrics of our instruments so that they can be used to really aid clinical infrastructure in myotonic dystrophy to research and plan future studies and make sure that a patient has a voice into the treatment's effectiveness. And so at the end of the day, we're really going to look to determine how patient-reported disease burden changes over time, identify participant preference for clinical trial measures, identify potential treatable factors with faster or slower disease progression, optimize patient-centered clinical trials tools, and the ultimate goal is to facilitate future therapies for patients who suffer with myotonic dystrophy type 2. So thank you for listening to that presentation. Really am grateful for the MDA and for our patients that we have the honor to work for, and like to thank our CHET team and our outcomes team who really have been instrumental in all the work that we've done. Thank you.
Thanks, Dr. Heatwole. It's really remarkable to see just the development of all these rigorous health indices across so many diseases. They're really making a difference for clinical trials. Tess, are there any questions in the chat from our audience for Dr. Heatwole at this time?
No specific questions for Dr. Heatwole at this time. We did have someone ask how they can get an appointment at the University of Rochester, so I think there's a lot of interest in what you're working on. Thank you, and Angela or Dr. Leck and I are answering questions in the chat as they come up in a general sense, but no specific questions at this time. Thanks.
Okay, so I guess we'll move on to Dr. Nolan and we'll field more questions at the end if there's time.
Okay, there is also questions in the main chat, not the Q&A.
Okay. So welcome, Dr. Nolan. Dr. Nolan is currently a postdoctoral fellow in Dr. Clotilde Laudier-Turen's lab at Mass General Hospital and Harvard Medical School. And prior to his postdoc role, Dr. Nolan earned his PhD in clinical neuroscience from the University of Oxford, and his research focuses on molecular mechanisms underlying ALS, and he'll tell you more about his specific research. So welcome, Dr. Nolan.
Thank you very much. Yeah, so my name's Matthew. I'm an instructor in neurology at Massachusetts General Hospital and Harvard Medical School. I've been here since 2019. Just after I finished my PhD, I moved to the US. I'm originally from London. And at the lab that I work as part of here, we broadly work on the role of RNA binding proteins and RNA binding protein homeostasis. But what that means is that we tend to focus on two diseases in particular, namely amyotrophic lateral sclerosis, or ALS, and frontotemporal dementia, or FTD. And I'm going to go into a bit about my work that I've done so far over the last 6 years, and then a bit about what our MDA-supported project is going to entail. So ALS, as a lot of people on the call might know, is a progressive neurodegenerative disease that involves the degeneration of both upper and lower motor neurons. So you have upper motor neurons that connect your motor cortex and your brain to your spinal cord, and lower motor neurons that connect your upper motor neurons, that connect your spinal cord to your muscles. And degeneration of both is required for the diagnosis of ALS. About 90% of cases are sporadic and there's no obvious genetic cause. About 10% of cases are familial, and the incidence of the disease can be traced through a family lineage. And that Stephen Hawking there was perhaps the most famous person who had the disease. And he was actually something of an outlier in that he managed to live for a really significant period of time after diagnosis. And there's limited numbers of FDA-approved treatments. So this is a very, very severe disease with limited treatment capacity. And it's really on us in the lab to understand the biology of the disease so that we can develop new treatments. Conversely, another disease, which is clinically very distinct frontal temporal dementia is a type of dementia and involves the degeneration of neurons within the frontal and temporal lobes of the brain. And this again is also a very severe disease. And perhaps the most famous sufferer now is Bruce Willis, who retired from public life a few years ago after progressively being less able to fulfill public engagement. So I mentioned that these two diseases were very clinically distinct, and that is true. One is a motor neuron disorder, and one is a brain type degeneration. But they're actually very closely related genetically and pathologically. To the extent now that we consider pure ALS and pure FTD to represent spectral extremes of disease. And some patients will develop a pure ALS syndrome. Some patients will develop a pure FTD syndrome. Some patients will actually go on to eventually be diagnosed with both diseases simultaneously. And there's the genetics of ALS and FTD are complex, but there are a lot of genes which cause both diseases, and there's some genes which cause either disease. So genetically, they're quite closely related. And also pathologically, they're also very closely related. So by pathologically, we mean, what does the brain look like post-mortem? What can we see under the microscope? And a majority of both diseases are underscored by the development of a pathology involving a protein known as TDP-43. And TDP is so important in both diseases that realistically, pretty much every lab around the world working on ALS and FTD are involved in something to do with TDP-43 research. It's that fundamental. And under normal conditions, TDP-43 is localized within the nucleus in the cell, which is where the DNA is stored, and it has functions in the nucleus and it does its thing. But under disease conditions, for reasons that we actually don't completely understand, that protein moves from the nucleus into the cytoplasm of the cell. And so you get a level of toxicity from that movement on two fronts. Firstly, you lose the normal function of TDP-43 from the nucleus, and you have this gain of function toxicity in the cytoplasm where the protein is, and it shouldn't be there, and it starts doing things to the cell which are detrimental. And in 2019, just before I moved to Boston and just before I started this job, this pair of papers was published back to back in Nature Neuroscience. One was from our group and Don Cleveland at UCSD, and the other one was from Kevin Egan at Harvard University. And both ultimately came to the same conclusion, which is that when TDP-43 is lost from the nucleus, the other gene that's most affected is this neuron-specific microtubule protein called stathmin-2. And that was very interesting because stathmin-2 is one of the most highly expressed genes in the central nervous system, one of the most highly expressed genes in neurons. So if you have this disruption to TDP-43 and you subsequently disrupt the expression of a gene which is very highly expressed in neurons, the idea was that this is likely to have a very significant negative effect on neurons. And it's particularly important because neurons have very long processes. Your motor neurons, for example, from your cortex to your spinal cord, extend their axons up to 1 meter in length. So they have a very big requirement for this gene, stathmin-2. And the fact that two groups independently came up with the same thing at the same time, that doesn't happen all the time. But it gives us confidence that, that this stathmin-2 may therefore be a therapeutic target applicable to both ALS and frontal temporal dementia. So I was hired kind of with the brief that my job was to look for ways that we could alter stathmin-2 expression. So whether that be promoting its expression and it be a therapeutic opportunity, or to negatively influence its expression in the hope that we might learn more about its biology. So I spent the first couple of years developing cellular tools and models that I was going to need to run subsequent screens. And we are now kind of on this bit on the right where we've identified lots of targets and genes and drugs, and we've spent the last few years validating their effect. And we've now submitted the manuscript for peer review. So we hope that submission process goes well. But this is kind of the broad overview of what my work has involved thus far. And it hasn't just been me. I work as part of a team. We work as part of collaborator groups. We have collaborators all over the United States, all over the world, and in industry. And so this kind of work cannot be done by any single person or any single lab alone. And it's really been a privilege to be involved in something like this, where I feel like we have the real opportunity of influencing patient care ultimately. So one of the screens that I ran was to identify ways that we could change stathmin-2 expression was a drug screen. So I screened about 11,000 drugs for their effect on stathmin-2. And that sounds like a lot, but actually in industry, when they run these kinds of screens, they screen on the order of like hundreds of thousands, millions of drugs. So 11,000 was actually kind of a fairly targeted screening approach. But what we found was that of those 11,000 drugs, 6 of the top 10 most potent compounds were statins, cholesterol-lowering statins. And this is absolutely not something that we would have been able to identify otherwise. It certainly wasn't a finding that we were expecting to see. This is not biology that we would have been able to link purely by reading the literature. So this was very surprising, but the fact that the top 6 of the top 10 hits of the screen were all from the same drug class was an encouraging finding. And it's really this phenomena that I've spent the last few years trying to understand. And so we went on to show that statins can promote stathmin-2 expression in the context of cells that have a mutation in TDP43, but also where we artificially repress TDP43. So there's various ways that we can modulate TDP expression. And when we also subsequently treat them with a statin, we can make stathmin-2 go up to the normal level. And statins have been around for a long time. These are drugs that have been approved for decades. They have known toxicity profiles. They've been used in millions of patients across a long, long time. And so there is actually quite a lot of epidemiological evidence associating statin usage with positive outcomes in neurodegenerative disease. That's mainly derived from Alzheimer's related work, where there's a very large number of patients, and so it's easier to reach statistical power in those kinds of studies. But there is now more evidence being produced that statins may be of benefit in diseases like ALS from some kind of cellular experiments, but also in mice. So this is kind of contextual evidence that maybe this beneficial effect of statins, which can be seen across large epidemiological studies, may be being produced by the effect of the statins on stathmin-2 in neurons. And so that was the biochemical effect of the statins that we were able to identify, but also the phenotypic effect of the statins. When I put the statin on a cell in a dish, what you get is a promotion of a neurite extension phenotype. So you can see here the cells are kind of clustered and they're round. but when you treat them with a statin, they begin to extend what look like neurites. So to have an effect both genetically, biochemically, and phenotypically, which may be a benefit in patients, this was super exciting. And understanding the way that that occurs is what we've spent a lot of time doing. So we have treated cells and then run genetic experiments to understand how the statins are influencing the genetics of the cells. And we can see a lot of genes that are very relevant from a kind of pro-regeneration perspective being upregulated after statin treatment. So this is kind of a graphical abstract that we've submitted with our paper. So we found that statins can promote statin 2 expression, and they promote neurite extension in our cells in a dish. And we think overall these findings highlight modulation of this pathway that statins affect as being a potential therapeutic target to rescue statin 2 expression in the context of TDP-43 related disease. So the next question and the basis of our MDA development grant application was, which genetic changes are responsible for the statin-mediated effect on neurons? And that's a kind of level above question from what we've answered so far. So broadly, we look at the relationships between genotypes and phenotypes. And the genotype is the genetic background of an organism. The phenotype is any observable characteristic of an organism. And they're obviously closely related. Your genes influence your behavior, your physicality. So they're clearly two very closely related concepts. But then when you add in another variable, i.e. treatments, it gets considerably more complex. And what we want to understand is, how does a drug treatment influence the genotype and influence the phenotype, and how do those things relate as one? And an approach to be able to do this, which is now being done almost every day around the world, is an approach called CRISPR screening. And CRISPR, you may have heard of, is a technique that allows you to make precise genetic edits in cells and in organisms. And you can use it to run a screen whereby you make precise genetic edits in lots of cells simultaneously, and then measure some kind of phenotype after the fact. And that allows you to look at genotype-phenotype relationships at scale. Because before this, one of the major ways that we were doing these kinds of experiments was looking at, we treat some cells, we do an experiment, and then we measure the expression of one gene or two genes. And that's completely valid. We still do that every day. But if you want to do it at scale and understand relationships on a genome-wide basis, screening of this kind becomes a very, very powerful tool. But even that has some limitations, because what we're looking at here is the kind of the bulk effect on cells. And the question then is, what if we want to look at the relationship between a precise genetic edit, a drug treatment, and a specific phenotype? We need to be able to look at things at more like the single cell level. And so a collaborator at the Broad Institute, which is a large genetics institute in Cambridge, in Boston, in 2019 developed a method called optical pooled screening. And optical pooled screening was a method that allows us to actually visualize genetic changes in the cells and combine that with visual phenotypic changes. And this is something that my boss, my PI, Clotilde Lagetren, has really spearheaded the utilization of this technology in the investigation of neurodegenerative disease. And we are among the first labs to be doing this kind of thing. But there are now, I know because when I go to conferences, I see people talk about it, there are now a lot of interest in industry to conduct screens in this kind of approach, because the possibilities for how you set up the screen, the phenotypes you look at, the genes you look at, the treatments you put on the cells is almost endless. It's a complex method, but the technology for this kind of thing is changing and being improved at such a crazy rate, that it's really, we're in the kind of in the golden age of genetic medicine right now, and it's really exciting to be a part of it. So the question for us and how it relates to the statins and statin 2 is, which genetic changes are responsible for the statin-mediated effect in neurons? And that's the basis of our MDA development grant. And I actually just want to say thank you very much to the MDA, because it means a great deal to be part of things like this. We're extremely grateful, and on a professional and personal level, it means a great deal that they would put their faith in us to be able to work on this. And so the idea for me is that we use this as a kind of transitional grant, and hopefully at some point in the future I'll be able to run my own lab. And we're also, of course, I have to mention that we're grateful to all our collaborators and everybody else that works in our lab. As I mentioned, you can't do this kind of stuff in isolation, and also all our other funders. We get a lot of support from patients and donors, and yeah, thank you all for sitting here and listening to me talk on a Wednesday morning. Thank you very much.
Yeah, thank you, Dr. Nolan. It's great to have you in our MDA research community, and really excited to see the progress in your development grant in the years to come. Tess, are there any questions specific to Dr. Nolan?
Not yet, but those questions might be rolling in as we come. We do have a couple questions that came in last minute for Dr. Heatwole, if he's able to answer those.
Dr. Heatwole, are you still with us?
I am.
Perfect.
Great. So we had one question. Is there a study similar to the one for DM2 being performed for DM1 as well?
Yeah, so there's many studies actually for myotonic dystrophy type 1. So we, as I mentioned, one of our first instruments that we did using this type of technology is for myotonic dystrophy type 1, and that was done about 20 years ago. And that instrument is really highlighted as the NIH as the only truly recommended measurement to measure disease burden in myotonic dystrophy trials. And so to that end, it's being used in many industry trials. I mentioned the DINE study that are longitudinal therapeutic studies, and we're gathering extensive data regarding kind of how it's able to track disease progression, and more importantly, how it's able to detect therapeutic gain in some instances. And so that there's a lot of data there, and we're looking at it real time, and it's really exciting actually. And for myotonic dystrophy type 2, also very important disease that the MDA has at a very high level and views at a very high level. It's just not as much research and work done in that field. And so we're trying to close the gap there and really advance clinical infrastructure in that field, so those patients can also get clinical trials and develop therapies for their conditions.
Wonderful. Thank you so much. The other question we had for you was how to enroll in the current trial.
Yeah, so, and I'll tell my patients this, the best way to do that is there's a website called clinicaltrials.gov, and you can go on there and you can put the disease, whatever it is that you're interested in, and there's filter criteria, actively recruiting, intervention, observational studies, whatever type of trial you're interested in. And it will list all of the registered trials, both nationally and internationally. You can look at, are they recruiting? Are they being done locally? Are they remote studies? and you can see. And for our study, again, we're probably going to get up and running hopefully within the next month. And so look on clinicaltrials.gov, look up Leopard DM2, and you'll be able to see you'll see the website and the contact information to enroll in that study.
Wonderful. Thank you so much. Such an exciting time. It really is. I don't know if I see any other questions. I know Dr. Leck has also been keeping tabs on the chat, so maybe she can chime in if she sees anything. But thank you so much, Dr. Heatwole. All right, let's just give it one more moment in case anyone has a question they're wanting to type in. And I do also want to remind everybody, if you had a question at registration that we didn't answer, or if you had a really specific question about your diagnosis, I would encourage you to reach out to our resource center. We will have that information in the chat as well as at the end of the presentation. So if we're not able to answer your specific questions, this is more general overview of the new research and exciting endeavors that MDA is involved in. But again, please reach out to our resource center because we would love to help and support you with your questions. All right, I think that's all for now. So let me go ahead and share my screen and we will wrap up our presentations today. I know we're a little bit over, but I want to thank everybody for sharing their time and for answering all those great questions. Thank you. And we'd again like to thank and recognize our sponsor of today's program, Biogen. Thank you for supporting these educational programs for the neuromuscular disease community. Some terrific resources have been shared in the chat today. MDA offers the following resources related to this topic. We encourage you to take a look at these and hope you find them helpful. We hope you will continue to join us for future educational programs. MDA's community education team offers a variety of learning opportunities from webinars like today's to print-ready resources and self-guided workshops. Here are just a few of the programs we have coming up, as well as some of our on-demand webinars that are always available online. To learn more, please visit mda.org/care/community-ed or scan the QR code on your screen. We would like to hear from you. What did you like about today's program? What can we do differently in the future? As Dr. Lin mentioned, we will be doing these programs bi-monthly for a while. So if you have any input, we would love to hear from you. Please fill out the brief survey that will pop up right after the webinar. Your feedback is important to us. And again, if you have any questions, you're looking for extra support or resources, please contact MDA's Resource Center at 1-833-275-6321 or via email at resourcecenter@mdausa.org. We'd again like to thank our program presenters today, and thank you for sharing your time and expertise with us, Dr. Sumner, Dr. Heatwole, and Dr. Nolan, as well as Dr. Lin for leading us through this discussion. This will conclude today's webinar. Again, thank you for joining us, and we look forward to seeing you again soon. Have a good day.